Good day. I have the pleasure of talking to you today on as part of my receiving the Junior Scientist Award. And first off, huge thanks to the association for bestowing this award upon me. I think in academic life, very few things are as satisfying as being recognized by your peers. And so as such, thank you very much. It truly is an honor. And so today I'm talking about a big topic and entitled the Decoding Pancreatic Cancer from Bench to Scope. And I know that pancreatic cancer plays a role in many surgical pathologists' day-to-day interactions in the lab. And this talk is a bit different than giving you a primer on what to do when you encounter challenging pancreatic cancer H&E situations. This is going a bit into the basic science of pancreatic cancer and what we have been able to learn over the past decade, and giving you a glimpse into how these findings will actually come to the surgical pathology bench much sooner than I think in many other solid tumor types. So I hope you find it helpful. I hope you find it useful, and to maybe have a little enticing for you to stay on the recording is that at the end I will utilize some of the learnings from the basic science aspect into helping to streamline pancreatic cyst diagnoses. And that will be the last part of the talk. So hope that will entice some of you to stay on the recording. I do have some disclosures. I receive some consulting fees and I sit on some scientific advisory boards. But importantly, none of the material presented here is related to any of the commercial entities I'm involved with. So I think ever since I've been a medical student, I've been fascinated by the pancreas. It's such an intriguing organ, an organ we don't often think about, but it's so central to us and central truly by its location. And if any of you have ever spent time in an operating room, you know that the Whipple procedure is one of the most challenging operative procedures there is, just given by the close proximity that the pancreas has with so many vital structures within the abdomen. And pancreatic cancer is associated with an automatic fear, and this is right because the majority of pancreatic ductal adenocarcinoma are in fact 95% that account for pancreatic cancer, and a minority is only for PNETs. And so today, I'll be talking predominantly, if not exclusively, about PDAC, and I will be using the terminology of pancreatic interchangeably, but I wanted to make the point that there is a subset of pancreatic cancers that have a much better prognosis and do not really fall under the same category than PDAC. So we're all familiar with the surgical specimen of a pancreatectomy, and it's always the same picture. You have sometimes this very nice pancreatic duct, and then the tumor starts. And given that the pancreas usually only has a diameter of 2 centimeters, the pancreatic tumor often grows and gets encompassing the entire organ. And we're all very familiar with the H&E picture of this sclerotic, cirrhotic type of stroma surrounding the abnormal epithelial cells. And this picture highlights one of the biggest problems that we encounter as surgical pathologists is where is it cancer as up here, and where is it an atrophic lobule as is down here. And this lecture will not really talk about the intricacies as mentioned about the H&E diagnoses. I'm happy to give that talk at a different venue, but this is really trying to understand how this picture is in the making when we look at the genomic setup of pancreatic cancer. And so this is a picture that my collaborator, Dr. Gallinger, has shared with me. He's a surgeon at UHN in Toronto, and this was an operation, a Whipple operation that took almost 13 hours. And it truly is a piece of art. There's over 6 different anastomoses of the vasculature structures here, and it's probably one of the nicest and cleanest operating fields I certainly have ever seen. But the problem with that is despite having negative margins achieved with that operation, is that the patient actually did pass away from the disease 9 months later. And so we don't get to see this kind of picture. We get to see this type of picture, which is the Whipple specimen. And arguably, whenever we talk about pancreatic cancer, it is one of the most fear-instilling specimens for our learners as to how to handle a Whipple specimen and how to deal with that. And now you can argue that why do we care so much about these specimens when patients succumb to their disease despite heroic efforts of removing tumors in such a fast fashion? And it's true that 80% of PDACs are actually very late stage. They're metastatic or locally advanced meaning that they have already gone outside of the pancreatic organ, have encased some of the vessels surrounding it, and in that stage are often not amenable to surgical resection. So the resectable PDACs are actually here in the light blue are one of the minority of the tumors that we see, and there's a huge focus currently within the research world to change that, and one of the ways obviously is earlier detection. And not surprisingly, when you look at the overall survival and you compare metastatic towards resectable, there's quite a dramatic difference. So to get the curative intent surgery, it has to be the main emphasis of actually detecting pancreatic cancer early on. And so pancreatic cancer is now the 4th most common cause for cancer-related death in Canada. There's over 5,000 deaths annually. And it's encompassing predominantly the higher stages. So this is showing you stage 3 upwards. And so this is the problem, is that if you take all stages together, the 5-year survival rate for pancreatic cancer is now hovering just around 10%. And so this problem is just going to confound it even further, because pancreatic cancer rates will double by 2030 in Canada. We know that pancreatic cancer has surpassed breast cancer to become the 3rd most common cancer death in the US, shown over here, and it's still sitting as the 4th one in Canada, but that is likely to change, because in 2030, as mentioned, we know that the cases will in all likelihood double. So pancreatic cancer is something that is going to stay with us for quite some time. And the factors that are affecting that high mortality and that increasing burden are the late detection, as mentioned. They're incredibly high recurrence rate, as you've heard me say, for the patient who underwent Whipple operation with the picture early on. There's resistance to treatment, and there's some risk factors and aging population, which we can't really do very much about it. And then an area that my research has really been focusing on is the lack of clinically relevant biomarkers, and the key word here being that they're clinically relevant. And so when we talk about pancreatic cancer, we inevitably have to talk about some of the chemotherapeutic options, and there have been advancements made, and you have in all likelihood heard that the latest medication combination that is being used is FOLFIRINOX, as well as gemcitabine with nab-paclitaxel. And you can see that this is the median overall survival at 11 months to 8.6 months, which is not a large time frame, especially if you compare, but this is larger if you compare it to as to what gemcitabine alone was being able to give you. So again, there's lots of challenges to overcome in just treating pancreatic cancer once it's detected. And then part of that is that not all PDACs are the same. There's a high heterogeneity intra as well as intertumoral in the molecular makeup. And this has a direct impact on the clinical outcome, the treatment stratification, and the novel therapy design. So where FOLFIRINOX might give you a median overall survival of 11 months in one patient, it might only give you 3 months in another patient. And again, the challenge here is, and that's where we come in as pathologists and guiding that treatment for our colleagues in oncology. We do know that some morphological pictures of PDAC are associated with a different outcome, and probably the most famous one is the undifferentiated PDAC with osteoclast-like giant cells. And this is a paper I was involved where we demonstrated that even if you identify this subtype of PDAC on an FNA or an FNB, you can demonstrate that this has a better survival than those features who do not have these osteoclast-like giant cells. So there are some subtypes that we can differentiate, but the majority of subtypes we cannot at present time differentiate on an H&E slide, and it really comes down to doing molecular in-depth analysis to understand what those subtypes are. So this slide is my attempt to really break down some of the hallmarks in the molecular field that in my mind contribute to the low overall 5-year survival rate. And we don't really have to go through all of them, but it's sufficing to say that they are different in each one of them, and not one alone will probably be the answer to understand on how to treat and overcome the high mortality associated with pancreatic cancer. So my research program has really been taking those 8 different features and drive biomarker discovery, stromal targeting, look into the metabolic plasticity, look at novel therapies, understand the role that immunotherapy might play in pancreatic cancer, and then specifically also inform neoadjuvant therapy. And if to just highlight and drive home the point again, this is pancreatic cancer over here in terms of survival. So if we can just shift this curve somewhat over here, closer to lung or colorectal or liver cancer, we will have achieved a lot and moved the yardstick for our patients in terms of outcome. And so this is the belief that clinically relevant PDAC subtypes are molecular profiles that are associated with outcome and treatment response. And this is the overarching principle that I and many others in the field have been really working on over the past decade. And a little sidebar, I've been very fortunate to be a co-founder and to this date a co-director of Pancreas Center BC. And we have really been trying to emphasize that patient care can only improve with clear integration of research and clinical care programs. And we are working towards facilitating those multidisciplinary team interactions and accelerate that knowledge translation for clinical impact. And as a pathologist, I find it very rewarding to play a very active role in that clinical program. And for those of you who are still thinking about choosing an area, I think if you find an area where you can have a huge impact as a pathologist on the clinical teams, those, in my opinion, are very rewarding careers. And I've been very fortunate to really, in Pancreas Center BC, highlight the translational research focus as well as the clinical care and the researcher engagement. And we have been very successful in changing established researchers' progress or research areas in other solid organ tumors to pancreatic cancer. And so what I'm presenting here today is by no means all my own research. This truly is a team effort, and Pancreas Center BC has played a huge role in all of that. And so when it comes to the pathology aspect, again, I believe that we can have a huge impact in the paradigm shift on how we deal with pancreatic cancer. And so for those of you who've gone to tumor boards where pancreatic cancer treatments get accessed, the majority, especially in Canada and in North America, is still that radiology assessment trumps most of the decision making. And that the treatment is based on that radiology, on the extent of the disease, and pathology often only has a confirmatory role in saying, yes, this is in fact tumor, which is important, but doesn't really change the way we could change treatment. So I believe that if we molecularly subtype pancreatic cancer up front, and we do not necessarily wait until the resection, we will be able to change how treatment decisions are being made up front, and we will be closer to the patient in deciding what treatment option to choose. So in order to do that, we really have to understand the genomic and transcriptomic landscape of PDAC. So the next couple of slides are just highlighting some of the challenges we're working on in pancreatic cancer, given that there is a very homogeneous expression of KRAS, but there's many other things that are happening that may inform how tumors behave. So what I'm showing you here are singlet nuclear variants and copy number variations in 100 PDAC. So the majority of them will have KRAS mutations, and only a minority are wild type for KRAS. But interestingly enough, those wild type for KRAS cases are a treasure chest for finding new and targetable targets. And one of the things that we found together with collaborators was an NRG1 fusion, which is now sensitive to afatinib, and I'll show you some of the results on that one. T53 is the next common mutation that we encounter in PDAC, and do note it's not always in conjunction with KRAS. Sometimes it's overlapping or concurrent. And then obviously CDKN2A is then coming down, and the last one is SMAD4, which has gained a bit of traction, especially in the pathology world, given that there's a good IHC marker. But as you can see here, it only really is expressed in 38% of pancreatic cancer. The other big aspect of the genomic landscape of PDAC is the chromosomal structural variations. And we know that there is the majority of cases have what we call a scattered chromosomal structure variation. And then there's stable, locally rearranged, and unstable. And all of these can have impact on the treatment decisions. And we know that if you have a high BRCA1/2 signature in your PDAC tumor, that you're very sensitive to a platinum-based chemotherapy. So again, it's not enough to just look at the single nuclear variants or the copy number variation. Chromosomal structure variation are very important for treatment, even right now in 2020, to understand which patients might respond to a platinum-based chemotherapy. And then the latest thing really is to understand the transcriptome of pancreatic cancer. And there's the simplified way, as has been demonstrated in other solid organ tumors, is to subdivide PDAC into basal-like or squamous quasi-mesenchymal subtypes and the classical one, and the basal-like being the minority one. And so if you take all of this together, those three ones, that really encompasses the molecular signature of pancreatic ductal adenocarcinoma. And the transcriptome-based subtypes are very intriguing because, in fact, the basal-like subtype is associated with the poor prognosis. And this has been demonstrated in multiple data sets now. And arguably, one of the more well-known one is the Moffitt one, which just really subdivided classical versus basal. And you can see that gap here in the Kaplan-Meier survival curve. And so just like in other solid tumors, we know that this basal-like expression on the molecular profile is associated with a poor prognosis. And this is a very nice figure taken out of a review paper from Collison and colleagues, and really going through as to what the pathway is to determine as to which one of the tumors will end up in the basal or in the classical subtypes. But I want to highlight to you that all of this data is really identified predominantly in primary resectable pancreatic ductal adenocarcinoma, and that poses a problem because, as I've told you earlier, the majority of pancreatic ductal adenocarcinoma are actually not resectable, but are metastatic, as demonstrated over here. So we make decisions on some of the subtyping based on a subset of tumors that are not representative of the overall clinical picture that we're dealing with. And so then what is the clinical relevance thereof? So we believe that understanding these transcriptomic subtypes, it may help us to personalize the treatment and develop novel therapies. We are very intrigued as to whether we can target specific subtypes. And we also want to understand what cellular functions or processes are associated with those distinct subtypes. And that really led us 3 years ago to initiate this research program, which is a national research program called APIC, standing for Enhanced Pancreatic Cancer Profiling for Individualized Care, and really get a large section of patients across Canada with pancreatic cancer. And at different sites, you see them listed over here, and subtype them accordingly. And subtype them not just based on genomic variants, but subtype them based on transcriptomic variants as well, to understand that really whole genome and whole transcriptome are the best way of understanding what the molecular subtypes are. And I recognize this is a very convoluted slide, but I wanted just to highlight the fact that this EPIC knowledge bank that we have been building, and by now we have close to 300 patients in it, has been intricately linked with some of the clinical trials, Canadian clinical trials group work. And so it really has allowed us to make almost real life decisions for patients, real-time decisions for patients as to how to change and approach the treatment, but it's also been very helpful for identifying new target strategies going forward. And EPIC really involves the integration of multiple PDAC data from multiple sequencing centers and platforms. And one of the early things that we realized is sequencing in one center is not the same as sequencing as another center. And this is really goes back to having different batches, which are demonstrated for you here. So there have been multiple run through it with the help of our very strong bioinformatics team, and having batch correction, which is done over here, so that we can actually compare data from one center to another, all with the ultimate goal is that what we find here, we might be able to distill down to the individual patient. And so one of the early success stories of this program has been, as I mentioned earlier, identifying the NRG1 fusion. And this was in KRAS wild type cases. And you can see 2 patients here on their PET scan having a very high load of metastatic disease, and 4 weeks after afatinib treatment, both of those patients had resolution on PET scanning. Now, they did have recurrence of the disease 2 months and 3 months respectively later, but it demonstrates the concept that we are able to target individual changes in the molecular makeup of pancreatic cancer, and gene fusions might be one of them that are targetable. And so we have now focused a lot of our work in understanding if there's any kind of fusions further in PDAC cancer that are targetable, but it again highlights the aspect that you have to have a whole genome and whole transcriptome sequencing to understand the intricacies associated with that. Another huge area that we've come to realize to play a huge role in pancreatic cancer is cell metabolism. And I'm showing you here the CAC pathway of cell metabolism. And arguably, glycolysis and cholesterol synthesis are key ingredients for most cancer subtypes. But it's become abundantly clear that they play a huge role in pancreatic ductal adenocarcinoma. And so we were able in our group to stratify PDAC based on the expression of glycolytic and cholesterol genes into really 4 distinct subtypes: a glycolytic subtype, a cholesterogenic subtypes, a quiescent subtypes, and a mixed subtypes. And we're able to do this in both a metastatic cohort of patients with pancreatic cancer and those who had resectable cancer. And in both of those patient cohorts, we could demonstrate that the glycolytic tumors were associated with a shorter overall survival. And this is intriguing because we also know that the KRAS and MYC amplifications were associated with an increased glycolytic signature. And as you know, that is the predominant genomic makeup of those tumors. So we believe that there is a very close link between the molecular signature as well as the metabolic signature. And these metabolic profiles are associated with prognostic subtypes. And again, they give us the potential to target those biological mechanisms for subtype differences. And this is just here to highlight to you that the basal subtypes and the classical subtypes might sometimes match the metabolic subtypes, but it's not, as one would hope, always a clear-cut overlap. And again, goes to show is that subtyping pancreatic cancer might take more than just looking at one specific subtype. So we believe that the clinical relevance of metabolic subtyping might lie in the biomarkers. And as mentioned earlier, there's very few clinically validated biomarkers. So these signatures may expose some of the vulnerabilities to agents targeting the metabolism. It obviously is intriguing to think that a glycolytic or basal-like tumor might be more sensitive to drugs targeting the glycolysis pathway. We're also thinking about tumor targeting the tumor metabolic phenotype with specific subtypes, and then thinking about a subtype switch. What if you could change a glycolytic subtype to a more cholesterogenic subtype, and thereby changing a basal-like into a classical subtype for better treatment response? And this is a real chance for us, because there is, these subtypes are associated with the time to recurrence, especially in the resected PDACs. So changing these subtypes and changing the makeup of the tumor might allow to actually intervene in real time with treatment decisions. And one of the ways we are exploring that is really using patient-derived tumor organoids. And organoids have become a very popular avenue to study cancers in many solid organ tumors, but in pancreatic cancer, we're using them as part of APIC to make almost these real-time decisions. And so as patients get their biopsy, we sequence it in the whole genome and whole transcriptome. We also look at the metabolic subtype, and then in parallel, we run organoid cultures really for characterization and chemosensitivity screening, and all of that feeds back into an overall knowledge bank. And here's some pictures of the organoids, and we have demonstrated that not only do they recapitulate the primary tumor on the H&E slide, but we can also demonstrate in IHC staining that we can actually show that there's a difference between different subtypes based on genomic sequencing. And GATA6, I'll get back to that, but GATA6 is probably our most promising immunohistochemistry marker to date to subdivide classical from basal-like subtypes in pancreatic cancer. And so we're getting more and more information on those patient-derived tumor organoids, and they're demonstrating more and more that when we sequence them, that they are recapitulating the primary tumor, and that they might be a very good preclinical tool for translational research to understand and test out treatment strategies for individual patients. And here I'm showing you that we can treat these organoid cultures with different chemotherapeutic agents and get good responses with good cell viability data, really demonstrating to pick the right treatment for the patient at that time. So what does all of this mean for day-to-day surgical pathology sign-out? This is the picture I showed you earlier. Do we have to change the way we approach pancreatic ductal adenocarcinoma right now? Do we have to stain them differently with IHC markers? Do we have to change our reporting? And so we want to get to a place where we can tell our oncologist to use different treatments for different patients. I think we all understand that the approach of just giving a single chemotherapeutic target for any PDAC patient is no longer applicable. But in order to get to this very detailed view as to how to change treatment, what can we do at the end of the pathology side to guide that treatment in the right direction? And this goes back to developing IHC markers, and this has been a huge focus of my research group. And one of the things we did early on was build multiple pancreatic tissue microarrays to really study the IHC markers in detail and be able to translate the whole genome and whole transcriptome data into protein expression and IHC data. And arguably the most or the best candidate to come out of all of this research is GATA6. And I just showed you a GATA6 IHC image on the organoids, and GATA6 in our hands and in hands of our collaborators really allows to subtype basal from classical pancreatic cancer lesions. And so while this is important, if you are at a center where people will take this information and translate it into changing chemotherapy, it is not quite ready yet for prime time in every surgical pathology lab. But I wanted to bring this to your attention because I do believe that in the very near future, GATA6 will become a staple of subtyping pancreatic ductal adenocarcinoma. Another thing that has been taken further is that a group has now identified a purist score, and the purist score is based on many of the genes that are surrounding GATA6. And so really understanding that epithelial to mesenchymal transformation in pancreatic ductal adenocarcinoma, and this is a nanostring-based subtyping tool, and we have been able to use that tool in really subdividing on FFPE specimens patients into classical subtypes as well as basal subtypes. So nanostring, again, is not available in every lab, and I recognize that, but again, it's testimony to the fact that we might be able at a stage where, at a minimum, when we want to understand, is it classical, is it basal, that FFPE specimens may be sufficient and allow us to do that in the near future in the majority of surgical pathology labs. And GATA6 is a good example as to how we identify IHC markers to allow to help in that quest. Now, it would be nice if all the tumors would read the textbooks and would stay within their respective subtypes, but we recently have been able to demonstrate that there are a subgroup of tumors that follow a discordant subtype. And this is part of the hesitation I have in recommending changing current day-to-day surgical practice, because I do believe there's still a lot to learn and to understand in identifying what the subtypes are in pancreatic cancer before it's ready for prime time in the surgical pathology lab. And this slide here, and I promised it's the last one in talking of predictive biomarkers, that was really our attempt in a large cohort of pancreatic cancers to understand if single markers for MMR, for PD-L1, or for SPARC will show any changes, and hint one, any changes in the outcome, and should these be markers we'll be doing on a regular basis. And the answer is really here is that the heat map is all over the map, and not one single marker was indicative of showing response in this cohort to gemcitabine. And really that a single marker alone wasn't sufficient, but even in combination, they might not give us a clear picture of which patient responds to, in this case, gemcitabine. So the lesson learned from that experiment is what's really is that in the current way of how we're dealing with SINAD of PDAC is that there is not one single marker or a combination of markers that is sufficient enough to perform on a regular basis given the treatment implications. So as promised, I wanted to use pancreatic cysts to demonstrate of some of the molecular diagnostic stratification that we've learned from PDAC and how we can deploy that in day-to-day life surgical pathology. And I think all of us are very familiar with the different subtypes of pancreatic cysts that occur, and pancreatic cysts are interesting to study because you can clearly separate them into those that have a potential for malignant transformation and those who are benign and may be left alone. And as you know, they lend themselves for beautiful gross pictures, and exam questions, because some of them are so easily identifiable as the subtype that they represent. But arguably, that is not what we deal with on a day-to-day basis. And with more and more endoscopic ultrasound and fine needle aspirates being available in centers, we're getting smaller and smaller samples, and we're getting cytology samples, which, as having trained as an anatomical pathologist, still cause me a certain level of anxiety. And so the important management questions that we have to deal with in pancreatic cyst is, number one, is it a neoplastic mucinous cyst? And specifically, can we subtype them into an IPMN or an MCM, a mucinous cystic neoplasm? Or then secondarily, is it a non-mucinous benign cyst, such as a serous cyst adenoma or a pseudocyst? Or is it potentially another malignant neoplasm that presents itself in a cystic format, such as a PNET or a solid pseudopapillary tumor, or even a cystic pancreatic ductal adenocarcinoma? And again, the molecular subtyping of pancreatic ductal cancer has informed as to how we subtype these lesions. And this is now a bit of a dated figure, but it's still my favorite figure when it comes to understanding pancreatic cyst lesions and how nicely we can subtype them. And you will see that solid pseudopapillary neoplasms have mutations in beta-catenin, and we use that as an IHC marker to actually identify those lesions. We know that serous cystic neoplasms fall within the VHL ones, and we know that 3P deletions may be very helpful in subtyping as serous cystic neoplasms. And then we know that in the malignant ones, which are down here, we have KRAS mutations, we have GNAS mutations more commonly in IPMNs, and P53 mutations more commonly in MCNs. So I often refer to this figure, and I hope if you haven't seen it, you find it useful as well. Now, there's pretty much no endoscopist who will do a fine needle aspirate during endoscopic ultrasound of a pancreatic cyst without sending a part of the sample for biochemistry. And we know that the cyst fluid value of CEA and amylase is very helpful in the interpretation. And we know that CEA values of greater than 800 nanograms per mil are very indicative of a neoplastic mucinous cyst. And we know that low CA values usually do not represent a neoplastic process. Now, there are caveats to this, and again, at another venue, we can go into different aspects that are associated with that, but I just wanted to highlight to you that low CEA values can be seen in pseudocysts, and it's one of the biggest pitfalls associated with CEA values. And then low amylase values usually are not associated with the pseudocyst. So if I sign out pancreatic cyst cytology, I always look for the biochemistry, because it helps me in guiding me in the right direction and aiding me in the diagnosis. But arguably, molecular subtyping of pancreatic cyst has come a long way. And this is one of the integral papers at the beginning, demonstrating that really GNAS and KRAS mutation had a very high specificity and sensitivity for mucinous differentiations. And this is one of the key things that we, as mentioned, need to separate is, can we really differentiate neoplastic from non-neoplastic pancreatic cysts in that setting? And so in my own institution, we are using an allelic discrimination assay to really assess these pancreatic cystic lesions, and it's now become standard of care in my institution, and it's done routinely for every pancreatic cyst. And so what do the results mean? So if we do detect a KRAS mutation, and I showed you here in the earlier slide is that we're looking at multiple KRAS mutations, but a KRAS mutation supports a neoplastic cyst, the key word here being support, but it does not distinguish an IPMN from an MCN, and it does not correlate with grade. And I think that last point was very important, especially to my clinical colleagues, to highlight in the beginning when we started this project, because there was an immediate reaction that if you find a KRAS mutation, you deal with a lesion that has a high risk of malignant transformation, and that is not necessarily the case. If you find a GNAS mutation, this also supports an IPMN more over an MCM, so we do use it in combination to, again, guide people in the right direction, but it does not, again, correlate with grade. And then an RNF43 mutation supports also a mucin assist, but again, does not distinguish IPN and MCM. And we did this at the beginning, but we've dropped it as a target because we can really handle most of it with KRAS and GNAS very well. And then it mentioned 3P deletions, if identifiable, supports SCN. The problem there simple being is what kind of methodology you're using for your molecular assessment, because as most of you know, the VHL gene is a very large gene, and if you do targeted PCR, that would be quite challenging to do. But if you have an NGS panel, it is certainly something to take into consideration. And then lastly, as mentioned, beta-catenin mutation support and SPPT. So where to from here? What are the next steps? So I think we can subtype pancreatic cancer in many different forms. Some of them we're very lucky that they might already respond to a targeted treatment. Some of them we can advise our clinical colleagues that they may be of a basal classical subtypes and may respond better to the current chemotherapy. And then there's a whole nother subtypes of those patients that have very aggressive tumors and that are associated with the metabolic subtype. And we can identify those more, work more on subtyping them and targeting them in some of the work that I've shown you. We have extended EPIC now to also include the resectable and locally advanced tumors, and very excited that this is part of the Terry Fox Research Institute Marathon of Hope. So we hope that we will get more and more understanding also of the resectable ones and lessons learned from the metastatic ones into the resectable patient cohort. But I think the long-term goal is really to target the heterogeneity associated with PDAC through that personalized therapy approach. And I'm showing you here a figure taken from a very great review article from about now 2 years ago, demonstrating that tumors compromise that very heterogeneous collection of cells. And all of these cells have their own distinct genetic and phenotypic properties that can differentially promote progression, metastasis, as well as drug resistance. And there are extrinsic factors that impact on the intermetastatic lesional subtype differences, and there are intrinsic factors that mediate the subtype and the plasticity of the tumor. So how do we bring all of these different and arguably very challenging aspects into the subtyping of PDAC, and how do we implement that into a clinical specimen subtyping? So I still believe that the FFPE is our friend in all of that, and that the classical pathology will help in the analysis thereof. Biomarkers certainly are coming down the pipeline, such as GATA6, but I also believe that pathologists play a key role in the targeted sequencing, not just in identifying the area to be sequenced, but also putting it in the right direction and pushing our colleagues in sequencing lesions that we do believe have the potential to show subtypes that may have a patient impact. And then lastly, we didn't really talk about very much about this, but liquid biopsies obviously in solid tumors play a huge role, but need to be correlated back to the tissue to be of meaningful importance. So my last slide is really an acknowledgement slide. This is a huge team effort, and really wanted to highlight 3 people in the Pancreas Center who are outstanding and who have helped tremendously in putting this together. This is Dr. Jana Kersinska, who's our senior scientist, Mr. Topham, who is our bioinformatician, and Mr. Calliger, who runs our tissue microarray. And then a huge thanks to my co-director of Pancreas BC, Dr. Renoff, who is always able to guide us in the right directions, and without whom a lot of this would have not been possible. So with that, thank you all very much. And I really appreciate, again, given the opportunity by the association to give this lecture as part of the Junior Scientist Award. And thank you very much for that.